Staining Cheek Cell Lab by Adelyn Hamrick. How do you stain cheek cells so you can later see them under a microscope? Let me show you. The needed materials for this lab are a microscope, toothpicks, napkin or paper towel, human cheek cells, wet mount slide, cover slip, and blue dye. Step 1. Grab a microscope, plug it in, and turn it on. Step 2. Grab a toothpick. Step 3. Gently scrape the inside of your cheek with a toothpick to get cheek cells on it. Step 4. Then place toothpick on napkin so other germs don't get on the cheek cells. Step 5. Grab wet mount slide and cover slip from station. Step 6. Clean the wet mount slide and cover slip with water to get previous markings off. Step 7. Dry the wet mount slide and cover slip with paper towel. Step 8. Place a small drop of water on the wet mount slide for a place for the cheek cells. Step 9. Rub the cheek cells from the toothpick into the water so the cheek cells can be placed onto the slide. Step 10. Grab the methylene and place 1 drop onto cheek cells to stain the cheek cells blue. Step 11. Let the cheek cells absorb the dye for 10 seconds. Step 12. After adding blue methylene, carefully place cover slip over dye. Try to prevent causing bubbles. Step 13. Place 1 drop of water on one side of the cover slip to later help drain some of the dye. Step 14. In order to drain some of the blue methylene, you will need to grab a small piece of paper towel and then later place paper towel on the opposite end of the water drop. This will allow some of the blue dye to be absorbed into the paper towel. Step 15. After all of that, place slide under microscope and examine the cheek cells with low, medium, and high power settings. Final step. Switch from low to medium to high power settings to examine the cells closely. Enjoy!